close your eyes for a moment and imagine yourself in a quiet place. Perhaps you're lying in a meadow on a warm summer afternoon, feeling the gentle weight of sunshine on your skin. The grass beneath you is soft, yielding to your form like nature's own mattress. Above you, clouds drift lazily across an infinite blue expanse, their shapes constantly shifting, never quite the same from one breath to the next. There's no hurry here, no urgency. Time moves differently in this space. Your breathing slows, deepens, becomes rhythmic like the tide. With each exhale, tension melts away from your shoulders, your jaw, your hands. You're safe. You're comfortable. You're exactly where you need to be. In this peaceful state, your mind is free to wander, to explore, to discover. And tonight, we're going to embark on a journey together through one of science's most enduring mysteries, a puzzle that flows through the veins of millions of people around the world, yet remains fundamentally unexplained. We'll travel through time, across continents, into the microscopic world of cells and proteins, and perhaps touch upon questions that reach into the very nature of what it means to be human. But we'll do so gently, letting the story unfold at its own pace, like that stream you might hear bubbling somewhere in the distance of your imagined meadow. So settle in, get comfortable, let your body relax completely, and prepare to discover the fascinating mystery of Rh-negative blood. The story of Rh-negative blood begins not in some ancient legend or mysterious origin, but in the methodical, painstaking work of scientists in the early 20th century. To understand this discovery, we must first step back to a time when blood transfusions were deadly gambles, when doctors understood that mixing blood between people could result in catastrophic reactions, but they didn't know why. Enter Karl Landsteiner, an Austrian-American physician born on June 14, 1868, in Baden-Baden, a suburb of Vienna. Landsteiner was a man driven by curiosity about the fundamental nature of biological substances. He possessed that rare combination of meticulous attention to detail and bold imaginative thinking that characterizes the greatest scientists. After completing his medical degree at the University of Vienna in 1891, he didn't immediately settle into clinical practice. Instead, he spent years studying chemistry with some of Europe's leading minds, working with Emil Fischer in Würzburg, Eugene Bamberger in Munich, and Arthur Hans Schinzerg. This foundation in chemistry would prove invaluable for his later work in understanding the complex interactions happening at the molecular level in blood. When Landsteiner returned to Vienna and joined the Pathological Anatomical Institute in 1897, he began investigating a medical mystery that had plagued physicians for generations. Blood transfusions, which seemed like such a logical treatment for patients who had lost significant amounts of blood, frequently resulted in shocking, often fatal reactions. Patients would develop fever, their blood would clump together inside their vessels, their kidneys would fail, and death would often follow. In the year 1900, Landsteiner made a crucial observation that others had overlooked. When he mixed blood samples from different individuals, sometimes the red blood cells would stick together in clumps, a process called agglutination. But this didn't happen randomly. There were patterns, consistent and reproducible. Through systematic experimentation, mixing blood serum from one person with red blood cells from another, Landsteiner identified 3 distinct blood groups, which he labeled A, B, and C. The C group would later be renamed O, from the German ohne, meaning without, because these cells lacked the antigens present in the other groups. A 4th group, AB, was discovered shortly thereafter by 2 of Landsteiner's students, Adriano Sterli and Alfred von de Castello. This breakthrough in 1901 revolutionized medicine. Suddenly, blood transfusions could be performed safely, provided doctors matched the donor's blood type with the recipient's. The first successful blood transfusion based on this knowledge was performed by Reuben Ottenberg at Mount Sinai Hospital in New York in 1907. For this monumental contribution to human health, Landsteiner was awarded the Nobel Prize in Physiology or Medicine in 1930. But Landsteiner wasn't finished unraveling the mysteries of blood. Throughout the 1920s and 30s, Working at the Rockefeller Institute in New York after emigrating from economically devastated post-World War I Austria, he continued his investigations. In 1927, he discovered additional blood group systems, the M, N, and P types. Yet even with all these blood groups identified, medical professionals still encountered occasional transfusion reactions in patients who appeared to have matching blood types according to the ABO system. Something else was at play, another factor hidden in the complexity of blood that science had not yet illuminated. The answer came in 1937 when Landsteiner, now 69 years old, was working with his colleague Alexander Wiener. They were conducting experiments with rhesus macaque monkeys, injecting the monkeys' blood into rabbits to produce antibodies. When they tested these antibodies against human blood samples, they made a startling discovery. The serum reacted with approximately 85% of human blood samples, causing agglutination, but showed no reaction with the remaining 15%. This meant there was another antigen present on the red blood cells of most people but absent in others. They named this the Rh factor, short for rhesus factor, after the monkeys that had helped them discover it. The medical implications of this discovery became immediately apparent. Patients who were Rh negative, lacking this protein on their red blood cells, could develop antibodies if they received Rh positive blood. These antibodies could cause severe, life-threatening reactions in subsequent transfusions. Even more significantly, Rh-negative mothers carrying Rh-positive babies could become sensitized to the fetal blood during pregnancy or delivery, producing antibodies that would attack the blood cells of future Rh-positive children, leading to a condition called hemolytic disease of the newborn. Landsteiner and Wiener published their findings in 1940 in a paper titled An Agglutinable Factor in Human Blood Recognized by Immune Sera for Rhesus Blood in the Proceedings of the Society for Experimental Biology and Medicine. This discovery would eventually earn Landsteiner a share of the 1946 Albert Lasker Clinical Medical Research Award, though sadly, he didn't live to receive it. Karl Landsteiner died on June 26, 1943 at the discovery of the Rh factor completed our understanding of the major blood group systems used in transfusion medicine today. It explained the remaining cases of transfusion reactions and provided crucial knowledge for managing pregnancies. Doctors developed screening protocols to identify Rh-negative pregnant women and monitor them for antibody development. Later, in 1968, a preventive treatment called Rho-D, immune globulin, commonly known as Rogam, became available, dramatically reducing the incidence of hemolytic disease of the newborn. But even as this practical medical knowledge was being applied to save countless lives, a deeper mystery was emerging, one that continues to puzzle scientists to this day. Why did this Rh-negative trait exist at all? Where did it come from? And why had it persisted in human populations despite appearing to offer no advantage and, in fact, presenting significant reproductive disadvantages before modern medical interventions? To truly grasp the enigma of Rh-negative blood, we need to descend into the microscopic realm, into the world of cells and proteins, where the differences between Rh-positive and Rh-negative individuals play out at a molecular level. This isn't just an abstract exercise in biology. Understanding these mechanisms helps illuminate why this trait is so puzzling from an evolutionary standpoint. Every drop of blood coursing through your veins is an extraordinary substance, a living tissue comprising billions of cells suspended in a liquid medium called plasma. The most numerous of these cells are the red blood cells, or erythrocytes, specialized for one crucial function: transporting oxygen from your lungs to every tissue in your body and carrying carbon dioxide back to be exhaled. An adult human has approximately 25 trillion red blood cells, and every second your bone marrow produces about 2 million new ones to replace those that age and die. The surface of each red blood cell is studded with proteins and carbohydrates that serve as markers, identifying that cell is belonging to you and providing information about its type and function. These surface molecules are like the identification badges and flags that might appear on a ship, declaring its nationality and purpose. Among these surface markers are the blood group antigens, the proteins that determine whether you're type AB, or O, and whether you're Rh positive or Rh negative. The Rh system is actually far more complex than a simple positive or negative designation might suggest. The system comprises over 50 different antigens, but 5 of them are considered the most significant: D, C, E, and E. Note that there are capital and lowercase versions of C and E, representing different variants of these antigens, but there's no lowercase d. The D antigen is by far the most important clinically because it's the most immunogenic, meaning it's most likely to provoke an immune response if encountered by someone who doesn't have it. When medical professionals talk about someone being Rh positive or Rh negative, they're specifically referring to the presence or absence of the D antigen. If you have the D antigen on your red blood cells, you're Rh positive. If you lack it, you're Rh negative. This seemingly simple binary difference arises from variations in a gene called RhD, located on the short arm of chromosome 1. Individuals who are Rh positive typically have an intact, functional RhD gene that produces the D protein, which gets embedded in the red blood cell membrane. This protein is substantial, comprising 417 amino acids and winding through the cell membrane multiple times, creating a complex three-dimensional structure. The exact biological function of this protein in normal physiology remains somewhat mysterious. Research suggests it may be involved in transporting ions or gases, particularly carbon dioxide and ammonia, across the cell membrane, or in maintaining the structural integrity of the red blood cell. Interestingly, Rh-like proteins have been found in organisms as diverse as bacteria, algae, and worms, all sharing a basic function related to gas transport, suggesting this protein family is ancient and evolutionarily conserved. In Rh-negative individuals, the situation is different. The most common cause of being Rh-negative, particularly in European populations, is a complete deletion of the RhD gene. The gene simply isn't there. It's been removed from the genome, likely through a process called non-allelic homologous recombination, where similar sequences on either side of the gene caused it to loop out and be lost during cell division. This deletion happened in the distant past, and has been inherited through countless generations. Without the RhD gene, no D protein is produced, no D antigen appears on the red blood cells, and the person is Rh negative. However, being Rh negative isn't always caused by a complete gene deletion. In some populations, particularly in African and Asian groups, individuals may have the RhD gene present but carrying mutations that prevent it from being expressed or cause it to produce an altered protein that doesn't function as a typical D antigen. There are also rare variants where people have partial D antigens, with some but not all of the epitopes that define the full D antigen. This complexity can occasionally lead to unexpected blood typing results and highlights that Rh status isn't quite as simple as a binary on-off switch, though it functions that way for most clinical purposes. The inheritance of Rh status follows standard Mendelian genetics. The D antigen is dominant, meaning you only need to inherit one copy of the RhD gene from one parent to be Rh positive. If both your parents are Rh negative, lacking the RhD gene entirely, you will definitely be Rh negative because you can't inherit what they don't have. If one parent is Rh negative and the other is Rh positive with 2 copies of the gene, all children will be Rh positive. If one parent is Rh negative and the other is Rh positive but carries only 1 copy of the gene, heterozygous, each child has a 50% chance of being Rh positive and a 50% chance of being Rh negative. The real medical significance of the Rh factor emerges when we consider what happens when an Rh negative person encounters Rh positive blood. The immune system is exquisitely designed to distinguish self from non-self, to recognize foreign molecules and mount a defense against them. When an Rh negative person's immune system encounters the D antigen for the first time, perhaps through a blood transfusion or during pregnancy, it recognizes this protein as foreign and begins producing antibodies against it. These anti-D antibodies are of the IgG class. immunoglobulins that are small enough to cross biological barriers. This is why they pose such a significant problem in pregnancy. An Rh-negative mother carrying an Rh-positive fetus can become sensitized to the fetal blood, particularly during delivery when some fetal blood cells typically enter the maternal circulation. Her immune system produces anti-D antibodies that remain in her bloodstream. In subsequent pregnancies with Rh-positive fetuses, these antibodies can cross the placenta, enter the fetal bloodstream, and attack the fetus's Rh-positive red blood cells. The resulting condition, hemolytic disease of the fetus and newborn, can range from mild to catastrophic. The antibodies bind to the fetal red blood cells, marking them for destruction. The fetus's spleen and liver work overtime to remove these antibody-coated cells and produce new ones. But if the destruction happens faster than replacement, severe anemia develops. The breakdown of red blood cells produces bilirubin, a yellow pigment that can accumulate to toxic levels, causing jaundice and potentially brain damage in a condition called kernicterus. In the most severe cases, called hydrops fetalis, the fetus develops total body swelling, heart failure, and fluid accumulation in body cavities. Before modern medicine, mortality rates for severely affected infants reached 20 to 40%. The introduction of Rho-D immune globulin in 1968 transformed this dire situation. This medication, given to Rh-negative mothers during pregnancy and after delivery of an Rh-positive baby, works by a clever mechanism. The administered anti-D antibodies quickly clear any fetal Rh-positive cells from the mother's circulation before her immune system can mount its own response and create lasting immunity. It's essentially giving the mother a temporary, passive immunity that prevents active, permanent sensitization. This simple intervention has saved countless infants and prevented immeasurable suffering. Yet this medical triumph also sharpens the evolutionary puzzle. For all of human history before 1968, before the development of exchange transfusions, before any medical interventions existed, Rh-negative women faced a significant reproductive disadvantage. Their Rh-positive babies, representing a substantial portion of their offspring, were at serious risk of death or severe disability. Historical data from before treatment was available suggested that approximately 1 in every 56 births to Rh-negative mothers resulted in infant death from hemolytic disease. Approximately 80% of Rh-negative individuals exposed to Rh-positive blood will develop anti-D antibodies, and once sensitized, the condition tends to worsen with each subsequent pregnancy. This brings us to the heart of the mystery. Natural selection is remarkably efficient at removing traits that decrease reproductive success. Genes that lead to fewer surviving offspring become less common over generations, eventually disappearing from the population unless balanced by some advantage. Yet Rh-negative blood hasn't disappeared. In fact, in some populations it's remarkably common. How did this trait persist and even flourish in the face of what should have been strong selective pressure against it? If you were to create a map showing the frequency of Rh-negative blood around the world, a striking pattern would emerge, one that deepens the mystery rather than solving it. The distribution is far from random, and the concentrations in certain populations demand explanation. Globally, approximately 6 to 7% of the world's population is Rh-negative, though some sources suggest the figure might be closer to 15% depending on the population surveyed. But this global average obscures dramatic regional variations that tell a more complex story. The trait is distinctly concentrated in certain areas while being virtually absent in others, suggesting a history of genetic isolation, founder effects, or perhaps selective pressures that varied by geography. Europe stands out immediately as the epicenter of Rh-negative blood. Across the continent, approximately 15 to 17% of people are Rh-negative, more than double the global average. But even within Europe, the distribution is uneven. Northern and Western European populations show higher frequencies than Southern and Eastern Europeans. In the United Kingdom, about 16% of the population is Rh-negative. In Scandinavia, the figure is similar. Moving south toward the Mediterranean, frequencies decrease somewhat but remain elevated compared to global averages. The story changes dramatically when we look at other parts of the world. In Asia, Rh-negative blood is remarkably rare. China and Japan report frequencies below 1%. In many Asian populations, being Rh-negative is so uncommon that individuals with this blood type may face challenges finding compatible blood for transfusions in medical emergencies. The indigenous populations of the Americas and Oceania show similarly low frequencies, often below 1%, suggesting that this trait was virtually absent in these populations before European contact. In Africa, the distribution is more complex and varied. Many sub-Saharan populations show Rh-negative frequencies of 1 to 3%, low but not absent. However, some North African populations display much higher frequencies, in some cases approaching 30%. This variation within the African continent is itself intriguing, hinting at different historical population movements and genetic influences. The Middle East presents an intermediate pattern, with Rh-negative frequencies generally higher than in Asia but lower than in Europe, typically ranging from 8 to 15% depending on the specific population. This gradient, decreasing as one moves from west to east, suggests a possible center of origin or expansion in the western regions. But the most extraordinary concentration of Rh-negative blood, the statistical outlier that has captured the imagination of researchers and conspiracy theorists alike, occurs in a small region straddling the Western Pyrenees Mountains between France and Spain. This is Basque Country, and here the frequency of Rh-negative blood reaches levels found nowhere else on Earth. Approximately 25 to 30% of ethnic Basques are Rh-negative, and in some isolated areas where the Basque language and culture are best preserved, the frequency has been reported as high as 35% or even higher. One careful genetic study found a frequency of 47.2% in their Basque sample, though this may represent a particularly isolated subset of the population. To put this in perspective, if you gathered 100 randomly selected Basques, approximately 30 of them would be Rh-negative. In a comparable sample from China, you might find 0 or 1. This dramatic concentration cannot be dismissed as statistical noise or sampling error. It represents a genuine biological phenomenon that demands explanation. What's particularly intriguing is that other areas known to have been refuges during the last ice age also show elevated frequencies of Rh-negative blood, though not as extreme as the Basque Country. The Atlas Mountains of Morocco, the Canary Islands, parts of Ireland and Scotland, Corsica, and Crete all show higher than average concentrations. This pattern has led researchers to speculate about connections between these populations and ancient migration routes or shared ancestry going back to prehistoric times. The geographic distribution pattern suggests several possibilities. One is that Rh-negative blood is an ancient trait that was more common in early European populations and has been diluted over time by migrations and mixing with other populations. The Basques, because of their geographic and linguistic isolation, have maintained this ancient genetic signature. Another possibility is that the Rhd gene deletion occurred multiple times independently in different populations, though this seems less likely given that genetic analysis suggests a common origin for the European variant. A third possibility, which we'll explore later, is that there has been some selective advantage to being Rh-negative in certain environments or under certain conditions, allowing the trait to increase in frequency despite the reproductive disadvantages. The distribution also has practical medical implications. Blood banks in different countries must maintain inventories that reflect their local population's blood type frequencies. In a country like China, where Rh-negative blood is extremely rare, maintaining adequate supplies of Rh-negative blood for the small percentage who need it can be challenging. The Rh-negative individuals in such populations may need to bank their own blood in advance of planned surgeries or may face difficulties in emergency situations. This global distribution pattern is like a genetic fingerprint, a record written in blood of human history, migrations, and survival. It tells us that populations have not simply mixed homogeneously across the globe but have maintained distinct genetic identities shaped by geography, culture, and historical accident. The question is, what story is this fingerprint telling us? Is it simply a tale of random genetic drift, of isolated populations maintaining a neutral trait by chance? Or does it point to something more? Perhaps to selective pressures we don't yet understand, or to a complex history of human movement and mixing that standard narratives have overlooked. To understand the concentration of Rh-negative blood in one place above all others, we must delve into the mystery of the Basque people themselves, for they are as enigmatic as the blood that flows through their veins. The Basques have long puzzled anthropologists, linguists, and geneticists, standing as a sort of living fossil, a people who seem to represent something very old, very distinct, preserved in the mountains of southwestern Europe like a message from the deep past. The Basque country, or Euskal Herria as it's called in the Basque language, occupies a relatively small territory in the Western Pyrenees. It encompasses 7 provinces, 3 in France and 4 in Spain, covering an area of roughly 20,000 square kilometers. The landscape is dramatic. Rugged mountains rising abruptly from the sea, deep valleys, swift-flowing rivers, and along the coast, picturesque fishing villages and sophisticated cities like Bilbao and San Sebastian. This geography has historically made the region somewhat isolated, not completely cut off but buffered from easy movement of large populations. What truly sets the Basques apart, however, is their language. Euskara, the Basque language, stands alone in Europe, utterly unlike any neighboring tongue. It is not an Indo-European language, meaning it shares no common ancestry with Spanish, French, English, German, or virtually any other European language. While most European languages show clear relationships, sharing vocabulary, grammatical structures, and sound patterns that reveal their common origins, Euskara is different. It's what linguists call a language isolate, a language with no known relatives. The structure of Euskara is complex and fascinatingly different from Indo-European languages. It uses a grammatical case system more elaborate than Latin, with multiple cases indicating different relationships between words. The language is agglutinative, meaning it builds complex words by adding suffix after suffix to roots, creating single words that might require entire phrases to translate. The word order differs from neighboring languages, and perhaps most intriguingly, the vocabulary contains words that seem to date from a time before agriculture, before the Bronze Age, perhaps to the Stone Age itself. Consider the words for tools and implements. Eitzer means hoe, iskora means axe, isto means knife, and various words for digging and cutting all derive from hates, or aites, meaning stone. This suggests a language that developed and fossilized when stone tools were the primary technology. Such etymological evidence indicates Euskara's extraordinary age, pointing to origins in the Paleolithic period, tens of thousands of years ago. Most European languages can be traced back 4,000 or 5,000 years to Proto-Indo-European. Euskara appears to reach back perhaps 5 or 10 times deeper into the past. This linguistic isolation parallels genetic findings. Genetic studies have consistently shown that Basques are distinct from surrounding populations. They show genetic markers suggesting their ancestors inhabited this region during the last glacial maximum, the peak of the last ice age approximately 20,000 years ago, when much of Europe was covered by glaciers and ice sheets. The Basque country, with its coastal location and relative mildness compared to areas further north, served as a refugium, a place where humans could survive while others were forced to retreat. As the ice retreated and Europe became habitable again, populations expanded from these refugia, repopulating the continent. But some people remained in the original refuge areas. The Basques appeared to be, at least partially, descendants of those who stayed, maintaining genetic and cultural continuity from Paleolithic times through the arrival of Neolithic farmers, through the Bronze Age, through successive waves of Celtic, Roman, Visigothic, and Moorish. The combination of linguistic isolation, genetic distinctiveness, and the unique blood type distribution has led to various theories about Basque origins. Some researchers have proposed that Basques are descendants of Cro-Magnon populations, the anatomically modern humans who arrived in Europe approximately 45,000 years ago and are known for their cave art and relatively sophisticated material culture. The idea is that as later populations moved into Europe, particularly during the Neolithic period when farming spread from the Middle East, these newcomers mixed with and largely replaced the older hunter-gatherer populations. But the Basques, protected by their mountains, resisted this displacement. Another theory connects the Basques to a broader pattern of genetic and linguistic survival in Ice Age refugia. According to this view, several distinct populations survived in different European refugia during the glacial period in Iberia, Italy, the Balkans, Ukraine. As the ice retreated, these populations expanded, but those in the refugium maintained higher concentrations of ancient genetic variants. The Basque country was one such refugium, and the Basques are its primary surviving representatives. The high frequency of Rh-negative blood in this population could be explained in several ways within these frameworks. If Rh-negative blood was common in Paleolithic European populations, the Basques have simply retained this ancient trait while other populations have been diluted by admixture with groups having different blood type frequencies. Alternatively, the Rhd gene deletion might have occurred specifically in the population ancestral to the Basques, and its high frequency there represents the founder effect, where a trait present in a small founding population reaches high frequencies in descendants simply through genetic drift, not because it provides any advantage. However, these explanations face challenges. Genetic drift typically requires very small population sizes to dramatically change gene frequencies, and while Basque populations were probably never enormous, there's no evidence they ever dwindled to the tiny sizes that would be necessary. Moreover, the reproductive disadvantage of Rh-negative blood should have slowly reduced its frequency, even in an isolated population, unless balanced by some advantage. Some researchers have therefore proposed that there must be a selective advantage to being Rh-negative under certain conditions, perhaps related to disease resistance or some other health benefit that we haven't yet identified. The Basques themselves have been aware of their uniqueness for centuries, maintaining a strong cultural identity despite pressure from surrounding states. They've resisted assimilation, maintaining their language, though it's now spoken by perhaps only a quarter of the population in the Basque regions, down from higher percentages in the past. Their traditional sports like pelota, their distinctive cuisine featuring ingredients from both mountains and sea, their traditional music and dance. There's a saying in Basque, Zazpiak bat, which means the seven are one, referring to the 7 provinces that make up the Basque country. This cultural cohesion has undoubtedly contributed to maintaining the genetic distinctiveness that researchers observe. Modern DNA studies have added nuance to our understanding. While Basques do show genetic markers of ancient European ancestry, they're not completely isolated relics. They do show some genetic input from Neolithic farmers who spread across Europe, and they've certainly experienced some gene flow from surrounding populations. But the proportions differ. Basques have more ancestry from Paleolithic hunter-gatherers and less from later agricultural populations than most other Europeans. They're not a pure line from the Stone Age, but they're the closest thing Europe has to one. The mystery of the Basques and their blood is part of a larger puzzle about human prehistory in Europe. Who were the first modern humans to arrive on the continent? How did they survive the Ice Age? What happened when farming populations arrived from the Middle East? How much mixing occurred? Which populations retained their distinctiveness and which were absorbed? The Basques stand at the intersection of all these questions, their blood a biological archive recording events from the deepest prehistory. If only we could fully read its message. Now we arrive at perhaps the most perplexing aspect of the Rh-negative mystery, the question that has troubled biologists since the trait was discovered and understood. How has this genetic variant survived and even flourished when it appears to offer no advantage and, in fact, presents a clear disadvantage under natural conditions? This is the evolutionary paradox at the heart of Rh-negative blood. a seeming violation of the fundamental principles of natural selection. To appreciate this paradox fully, we need to understand how natural selection typically works. Evolution by natural selection is deceptively simple in principle but tremendously powerful in practice. Organisms vary in their traits due to genetic differences. These trait variations affect the organism's ability to survive and reproduce. Those with traits that enhance survival and reproduction are more likely to pass their genes to the next generation. Over time, advantageous traits become more common in the population while disadvantageous traits become rarer and may disappear entirely. This process has shaped all life on Earth, refining and adapting species to their environments over billions of years. Traits that directly reduce reproductive success face particularly strong selective pressure. If a genetic variant causes individuals to have fewer offspring who survive to reproductive age, that variant should diminish in frequency each generation until it's eliminated from the population. The math is straightforward and unforgiving. Even a small reduction in reproductive success, say 10% fewer surviving offspring, will dramatically reduce a trait's frequency over time. Rh-negative blood presents exactly this kind of reproductive disadvantage. Before modern medical intervention, Rh-negative women faced significant risks to their reproductive success. Let's walk through the scenario in detail to see why this is so problematic for evolutionary theory. Consider an Rh-negative woman in a population where 85% of people are Rh positive, which is typical for European populations. When she becomes pregnant, there's an 85% chance her partner will be Rh positive, and if he is, there's a high likelihood the baby will be Rh positive, inheriting the dominant Rh positive gene from the father. Her first Rh positive pregnancy will usually proceed normally. The placenta acts as a barrier, separating maternal and fetal blood circulation. However, during delivery, some fetal blood almost inevitably enters the mother's circulation as the placenta separates from the uterine wall. The mother's immune system encounters the Rh-positive fetal red blood cell.
In approximately 80% of cases, the mother's immune system will respond by producing anti-D antibodies. She's now sensitized. These antibodies persist in her bloodstream for life. If she becomes pregnant again with another Rh-positive fetus, the antibodies cross the placenta and attack the fetal red blood cells. The fetus develops hemolytic disease, with its red blood cells being destroyed faster than they can be replaced. The condition typically worsens with each subsequent pregnancy as the mother's antibody response becomes more robust. Historical data from before medical interventions became available in the 1940s suggests mortality rates of 20 to 40% for affected infants, though some estimates place it lower, around 15 to 20%. Even infants who survived often suffered from severe anemia, jaundice, and potential brain damage from kernicterus. These complications would have reduced survival rates and overall fitness even among infants who lived through the neonatal period. A study from 1947 reported that approximately 1 in 56 births to Rh-negative mothers resulted in infant death from hemolytic disease. Over a reproductive lifetime, if an Rh-negative woman had 4 or 5 pregnancies, as was typical before modern contraception, she would likely lose 1 or more infants to this condition. Her effective reproductive output would be reduced compared to Rh-positive women who didn't face this hazard. This reproductive disadvantage should have caused the Rh-negative trait to become steadily less common over time. With each generation, fewer Rh-negative genes would be passed on because Rh-negative mothers had fewer surviving offspring. Mathematically, under these conditions, the Rh-negative trait should eventually disappear from populations, maintained only by the rare occurrence of new Rhd gene deletions, which would themselves be quickly selected against. Yet this hasn't happened. Not only has Rh-negative blood persisted, but in some populations, it's reached remarkably high frequencies. In the Basque Country, approximately 30% of the population is Rh-negative. This is more than simply the trait hanging on despite selective pressure. It suggests something else is going on. The evolutionary paradox becomes even more acute when we consider the heterozygotes, people who carry one copy of the deleted RHD gene and one intact copy. These individuals are Rh positive, because the presence of even one functional RHD gene is sufficient to produce the D antigen. However, when two heterozygotes mate, 1/4 of their offspring will be Rh negative, inheriting the deleted gene from both parents. Hemolytic disease of the newborn affects the Rh positive children born to Rh negative mothers, and these children represent both RHD gene variants. Each infant death removes one copy of the deleted gene and one copy of the intact gene. This creates an unstable equilibrium. The disadvantage falls on heterozygotes, and their loss affects both allele frequencies. Standard evolutionary theory suggests that under such circumstances, the rarer variant should be eliminated, though the process might take many generations. The fact that we observe Rh negative blood at considerable frequencies, especially in certain populations, suggests either that the selective pressure hasn't been operating long enough, or that there are counterbalancing forces we haven't fully identified. Several potential explanations have been proposed to resolve this paradox, though none has been definitively proven and the question remains open. One possibility is that the reproductive disadvantage of Rh negative blood is a relatively recent development in evolutionary terms. If the trait arose within the last few thousand years, there may not have been sufficient time for selection to eliminate it, especially in isolated populations where genetic drift can play a larger role than selection in determining gene frequencies. However, genetic evidence suggests the RHD gene deletion is actually quite old. Studies examining genetic diversity around the deleted region suggest it occurred tens of thousands of years ago, providing ample time for selection to have acted. This timing means the deletion likely predates the expansion of modern humans into Europe and was present in founding populations. If this is correct, there's been plenty of time for natural selection to work, making the persistence of the trait even more puzzling. Another hypothesis is that Rh negative blood provides some selective advantage that compensates for the reproductive disadvantage. Perhaps Rh negative individuals have enhanced resistance to certain diseases, or better outcomes in certain environments, or some physiological advantage we haven't yet identified. This kind of balancing selection can maintain genetic variance in populations even when they carry costs. The classic example is the sickle cell trait in regions where malaria is endemic. Individuals with 2 copies of the sickle cell gene develop severe anemia, but those with 1 copy gain significant protection against malaria, and this advantage in malaria-endemic regions maintains the allele in the population despite its costs. Could something similar be happening with Rh negative blood? Some research has suggested possible health differences between Rh positive and Rh negative individuals, though findings have been inconsistent and often controversial. Some studies have found associations between Rh status and certain diseases or conditions, but these associations are often weak and not replicated consistently across different populations. We'll explore these potential health connections in more detail later, but so far no clear, powerful advantage to being Rh negative has been established that would obviously outweigh the reproductive costs. A third possibility is that the reproductive disadvantage of Rh negative blood was mitigated under ancestral conditions in ways we might not fully appreciate. Perhaps ancient social structures, birth spacing patterns, or other factors reduced the impact of hemolytic disease. If pregnancies were more widely spaced, with longer intervals of breastfeeding between births, mothers might have fewer total pregnancies and fewer affected children. If infant mortality from all causes was very high, as it certainly was in prehistoric times, the additional mortality from hemolytic disease might have been proportionally less significant to overall reproductive success. Additionally, not all Rh negative mothers become sensitized, and among those who do, not all affected pregnancies result in severe disease or infant death. There's variability in immune response, in the degree of antibody production, in how severely the fetal cells are affected, and in whether the fetus can compensate for the cell destruction. Under prehistoric conditions, perhaps only the most severe cases resulted in infant loss, while less severe cases were survived, leaving overall reproductive success less impacted than we might calculate from modern clinical data. There's also the possibility that the observed frequencies are maintained partly by gene flow from populations where Rh-negative blood is maintained by drift rather than selection. If isolated populations maintained high frequencies of Rh-negative blood simply through random chance in small populations, and then intermittently contributed genes to larger populations through migration, the overall frequency might be maintained even if selection against it was operating in the larger population. Yet another intriguing possibility, though speculative, is that the heterozygote carriers of one deleted Rhd gene might enjoy some advantage that maintains the allele in the population. These individuals are Rh positive and don't face the reproductive penalties, but they carry the deletion and can pass it on. If having one copy of the deletion provided some benefit, even a small one, it could maintain the allele in the population despite the costs to homozygous Rh-negative individuals. This is analogous to the situation with sickle cell, where heterozygotes enjoy malaria protection without suffering severe anemia. However, what advantage a single Rhd deletion might confer is entirely unclear, and this remains purely hypothetical. The evolutionary paradox is complicated further by the observation that the most severe consequences of Rh incompatibility typically affect second and subsequent pregnancies. In evolutionary terms, reproductive success isn't just about the total number of offspring but about how many of those offspring survive to reproduce themselves. If an Rh-negative woman's firstborn survives, and perhaps her second, but later children are lost, she still achieved some reproductive success. From an evolutionary perspective, having two successful offspring is significantly better than having none, even if it's worse than having 5 or 6. Natural selection might not act as strongly against a trait that reduces but doesn't eliminate reproductive success, compared to one that causes complete sterility or death before reproductive age. We must also consider that human reproductive patterns have changed dramatically over time. For most of human history, we lived in small groups with relatively limited mate choice. In small populations, genetic drift, the random fluctuation of gene frequencies, can be more important than selection in determining which traits persist. If a small band of prehistoric humans happened to have a high frequency of Rh-negative individuals by chance, that frequency might persist through their descendants even without any selective advantage, simply because the group remained small and isolated enough that random events shaped their gene pool as much as natural selection did. The invention of agriculture, the growth of cities, the expansion of population sizes, and increased mixing between previously isolated groups have all altered the evolutionary landscape for humans. Traits that might have been maintained by drift in small groups would be subject to selection in larger populations. The fact that Rh-negative blood hasn't been eliminated even after these changes suggests either that these demographic transitions are too recent in evolutionary terms for selection to have completed its work, or that there really is something maintaining this trait beyond simple persistence of an ancestral variant. The mystery is deepened by the specificity of the distribution. If Rh-negative blood were simply a neutral trait maintained by drift, we'd expect to see it randomly distributed at low frequencies across all populations. Instead, we see a clear geographic pattern concentrated in Europe, nearly absent in Asia, rare in Africa except certain populations, and with a dramatic peak in the Basque Country. This pattern suggests a complex history involving population movements, isolation, mixing, and possibly selection acting differently in different environments. One scholar proposed that the high frequency of Rh-negative blood in Europe might be maintained by a historical pattern of maternal mortality. If pregnancy itself was extremely dangerous for women due to infections, complications, and other causes, and if this mortality risk meant that women rarely survived enough successful pregnancies to experience the full effects of Rh sensitization, the trait might persist because its costs never fully manifested. However, this explanation requires fairly extreme assumptions about maternal mortality that may not match historical or archaeological evidence. The evolutionary paradox of Rh-negative blood remains, to this day, an open question in human biology. It stands as a reminder that evolution is more complex than simple textbook descriptions sometimes suggest, and that real biological systems often involve trade-offs, historical contingencies, and factors we don't yet understand. The trait challenges us to think more deeply about human prehistory, about how populations evolved in isolation and then mixed, about the relationship between genetic and cultural evolution, and about the complex ways that selection, drift, and migration interact to shape the patterns we observe today. Perhaps most fundamentally, it reminds us that there's still so much we don't know about our own biology and history. Despite the sophistication of modern genetics, despite our ability to sequence entire genomes and track individual genes across populations and through time, this basic question, why has Rh-negative blood persisted, remains unanswered. It's a humbling reminder that nature still holds secrets, that the past still speaks to us in voices we haven't fully learned to interpret. If we're to solve the mystery of Rh-negative blood, we must venture back through the corridors of time, attempting to understand where this trait came from and when it first appeared. This journey takes us to the very edges of what can be known through genetic and archaeological evidence, into the murky realm where science meets informed speculation, where solid data gives way to competing theories and provocative hypotheses. The fundamental question is deceptively simple. When did the RHD gene deletion occur? Was it recent, within the last few thousand years, or ancient, dating back tens of thousands of years? Did it happen once or multiple times independently? Was it present in the populations that first evolved into modern humans, or did it arise later? These questions matter because they help us understand whether this trait has been shaped by natural selection over a long period or whether it's simply too recent to have been significantly affected by evolutionary pressures. Genetic analysis provides some answers, though not complete ones. By examining the DNA sequences around the RHD gene in people who are Rh negative, researchers can look for patterns that reveal the deletion's history. When a genetic change first occurs, it appears on a specific genetic background, a particular pattern of nearby genetic variants. Over time, recombination shuffles genes, breaking up these patterns and distributing the deletion onto different backgrounds. By examining how much variation exists around the RHD deletion, scientists can estimate how old it is. More variation suggests more time has passed since the original deletion event. Studies using this approach have suggested that the RHD deletion in Europeans occurred approximately 30,000 to 50,000 years ago, though estimates vary depending on assumptions about mutation rates and population history. This timing would place the deletion in the Upper Paleolithic period, the era when anatomically modern humans were spreading across Europe, developing sophisticated stone tool technologies, creating remarkable cave art, and competing and eventually coexisting with or displacing Neanderthals. This ancient origin has profound implications. It means the trait was present long before the development of agriculture, before the rise of civilizations, before most of recorded history. It places Rh-negative blood in the genetic toolkit of the first Europeans, part of the genetic diversity that characterized early modern human populations as they adapted to Ice Age Europe. But who exactly were these first Europeans? This question has been revolutionized in recent years by ancient DNA studies. Scientists have extracted and sequenced DNA from bones and teeth of people who lived and died thousands of years ago, creating genetic snapshots of prehistoric populations. These studies have revealed a more complex picture of European prehistory than previously imagined, with multiple distinct populations contributing to modern European ancestry. The story begins approximately 45,000 years ago when anatomically modern humans, Homo sapiens, arrived in Europe. They encountered a landscape already occupied by Neanderthals, a different human species that had inhabited Europe and parts of Asia for hundreds of thousands of years. For several thousand years, modern humans and Neanderthals coexisted, and we now know they interbred. Most people of European and Asian descent carry approximately 2 to 3% Neanderthal DNA, genetic material inherited from these long-ago encounters. Could Rh-negative blood have come from Neanderthals? This is one of the most persistent questions in popular discussions of the trait. The answer appears to be no. Studies of Neanderthal genomes have revealed which genetic variants were present in Neanderthals, and the pattern of Rh-related genes in Neanderthals appears to be consistent with them having been Rh positive. While there's still much to learn as more Neanderthal genomes are sequenced, the current evidence doesn't support a Neanderthal origin for Rh-negative blood. The populations that established themselves in Europe during the late Paleolithic are sometimes referred to as Cro-Magnons, though this term is somewhat old-fashioned and imprecise. These were anatomically modern humans, indistinguishable from modern people in their skeletal structure. They were sophisticated hunter-gatherers who created tools, jewelry, art, and apparently complex social structures. They survived the last glacial maximum, the peak of the last ice age approximately 20,000 to 26,000 years ago, when ice sheets covered much of northern Europe and survival required extraordinary adaptations. During the glacial period, humans in Europe were pushed into refugia, areas that remained habitable despite the harsh conditions. The most important of these refugia were in southern Europe, in the Iberian Peninsula, modern Spain and Portugal, Italy, the Balkans, and perhaps areas around the Black Sea. The Basque Country on the northern Iberian Peninsula with Atlantic coastal influences was part of this refuge area. For thousands of years, these isolated populations survived, and their genes were shaped by drift, by selection for traits suited to Ice Age conditions, and by the accidents of small population history. When the ice retreated, beginning around 19,000 years ago and accelerating after 15,000 years ago, Europe became habitable again. Populations expanded from the refugia, moving north and east, recolonizing territories that had been covered in glaciers or frozen tundra. The Basque Country was positioned at the western edge of this expansion, and populations spreading from there would have carried their genetic variants, including possibly high frequencies of Rh-negative blood, into newly available territories. This scenario would explain both the age of the Rhd deletion and its concentration in Europe, particularly in areas like the Basque Country that were refugia or close to them. The deletion might have been one variant present at moderate frequency in Paleolithic European populations generally, but it reached especially high frequency in the Iberian refugium by chance, and that high frequency has been partially maintained in the Basque population due to their relative isolation. However, this Paleolithic European origin is just one theory. The story was complicated by the arrival of new populations. Starting around 8,500 years ago, farming peoples began spreading into Europe from Anatolia, modern Turkey, and the Near East. These Neolithic farmers brought agriculture, pottery, domestic animals, and their own genetic variants. Over a period of several thousand years, they expanded across Europe, mixing with the existing hunter-gatherer populations in complex ways. Ancient DNA studies show that these incoming farmers were genetically distinct from the Paleolithic European hunter-gatherers. They had different ancestry, different physical characteristics, and presumably different languages and cultures. As they spread, they didn't completely replace the existing populations but rather mixed with them in varying proportions. In some areas, the genetic contribution of farmers became dominant. In others, hunter-gatherer ancestry remained substantial. The Basques once again appear distinctive. They have less farmer ancestry than most other European populations, suggesting they were less affected by this Neolithic demographic transformation. This fits with the idea that they maintain more genetic continuity from Paleolithic times. If Rh-negative blood was common in Paleolithic Europeans and less common in incoming farmers, the Basque retention of high frequencies would make sense. But there's another twist. Approximately 5,000 years ago, yet another population movement occurred. Peoples from the Pontic steppe, the grasslands north of the Black and Caspian Seas, began moving into Europe. These Bronze Age migrants, often associated with the Yamnaya culture, brought horses, wheeled vehicles, new languages. They appear to be the source of Indo-European languages that now dominate Europe and much of Asia, and again their own genetic signature. Their expansion was dramatic and swift, and they came to form a major component of ancestry in Northern and Central European populations. Interestingly, the Basques appear to have been less affected by this migration as well. They show relatively little steppe ancestry compared to their neighbors, and they retained their non-Indo-European language. Once again, geographic position and possibly cultural resistance helped them maintain genetic distinctiveness. This complex history of multiple population layers, each mixing with but not completely replacing previous inhabitants, means that modern Europeans carry ancestry from at least three major sources: Paleolithic hunter-gatherers, Neolithic farmers from the Near East, and Bronze Age pastoralists from the steppe. The proportions vary by region and population, creating the genetic diversity we observe today. Rh-negative blood, if it was indeed present in the Paleolithic hunter-gatherers and less common in the later incoming groups, would be expected to be most frequent in populations with the most hunter-gatherer ancestry, which is indeed what we observe. There's also the question of whether the Rhd deletion occurred only once or multiple times. Genetic analysis suggests that the most common form of Rhd deletion in Europeans is identical by descent, meaning all Europeans who carry it inherited it from a common ancestor who experienced the original deletion event. This supports a single origin, at least for the European variant. However, Rh-negative individuals in African and Asian populations sometimes show different molecular bases for their Rh-negative status, including different mutations or partial deletions rather than the complete gene deletion seen in Europeans. This suggests that the Rh-negative condition has arisen independently multiple times in different populations, though the complete deletion leading to Rh-negative status appears to be a single event that spread through European populations. Some researchers have proposed that the deletion might have occurred even earlier, perhaps in African populations before the out-of-Africa migration that gave rise to all modern non-African populations. If this were the case, the deletion would have been carried by the founding populations that left Africa and spread across the world. However, the very low frequencies of Rh-negative blood in most non-European populations argues against this scenario. If the deletion was present in the founding population, we'd expect to see it at least at low frequencies everywhere, unless it was selected against more strongly in non-European environments, which is possible but would require explanation. Another avenue of investigation involves looking at our more distant relatives. Studies have examined Rh-like genes in other primates, apes, and more distant mammals to understand the evolutionary history of this gene family. These studies show that Rh genes are ancient and highly conserved, meaning they've been maintained with relatively little change across millions of years of evolution. This conservation usually indicates that the gene performs an important function that is essential for survival. Yet the fact that some humans completely lack the Rhd gene and suffer no apparent consequences except the Rh incompatibility problem complicates this interpretation. Perhaps the function performed by the D protein can be compensated for by the related C and E proteins, or perhaps its function is important only under certain conditions. The deep conservation of Rh genes in mammals and other vertebrates does suggest that the capacity to lose the D protein is unusual and might represent a specifically human or primate adaptation. Most mammals appear to have the equivalent genes intact and functional. The loss of this gene in some human populations might be connected to other uniquely human features, perhaps related to immune system changes, dietary shifts, or other aspects of our evolutionary history. Some researchers have speculated about connections between Rh-negative blood and more exotic possibilities. Could this trait indicate interbreeding with other hominin species now extinct? We know modern humans interbred with Neanderthals and with another group called Denisovans in Asia. Could there have been others? While this is theoretically possible, there's no specific evidence supporting it, and the age estimates for the RHD deletion don't require invoking unknown hominins. The trait can be explained within the context of modern human evolution, even if all the details aren't yet clear. The search for the origins of Rh-negative blood is ongoing, with new techniques and discoveries continuing to refine our understanding. Ancient DNA technology improves every year, allowing extraction and sequencing of older and more degraded samples. Larger data sets of modern genetic variation are being compiled, enabling more sophisticated analyses of when and where genetic variants arose. Computational models of population history are becoming more refined, able to test different scenarios and determine which best fits the observed data. What emerges from all this research is a picture of Rh-negative blood as an ancient variant, probably arising in Europe or Western Asia sometime between 30 and 50,000 years ago, carried by hunter-gatherer populations through the Ice Age, concentrated by chance and isolation in certain refugia like the Basque Country, and persisting into modern times despite reproductive disadvantages that we still don't fully understand. It's a genetic echo of our deep past, a reminder carried in the blood of millions that our history is written not just in books and artifacts but in the molecular structures that make us who we are.
While genetic analysis provides one window into the past, archaeology offers another, grounding our understanding in the physical remnants of ancient lives. Though we cannot directly test the blood types of people who lived thousands of years ago from their skeletal remains, the archaeological record does provide context for the populations we've been discussing, helping us understand who these people were, how they lived, and how their cultures changed over time. The Upper Paleolithic period in Europe, spanning from approximately 45,000 to 10,000 years ago, represents the time when the RHD deletion likely occurred and began spreading through populations. This was an extraordinary era in human history. The people who lived during this time created the first unambiguous art, painting animals on cave walls with stunning realism and skill. Sites like Lascaux in France, Altamira in Spain, and Chauvet Cave contain masterpieces created 15 to 30,000 years ago, depicting horses, bison, aurochs, mammoths, and human hands, giving us a glimpse into the minds and lives of these ancient artists. The cave paintings found in the Franco-Cantabrian region, which includes the Basque Country and surrounding areas, are particularly numerous and spectacular. This concentration of Paleolithic art coincides geographically with high frequencies of Rh-negative blood in modern populations, though whether this connection is meaningful or coincidental isn't clear. The artists who created these images were anatomically modern humans, with brains and bodies essentially identical to ours. They were our ancestors, and if current theories are correct, many carried the genetic variant we're investigating. Archaeological evidence from this period reveals sophisticated hunter-gatherers who used a wide variety of tools, including blades, scrapers, burins for engraving, harpoons for fishing, and spear throwers called otlotls that extended the range and power of thrown projectiles. They made bone needles indicating tailored clothing. They created jewelry from shells, teeth, and carved bone and ivory. They buried their dead with grave goods, suggesting belief in an afterlife or at least complex ritual behavior. The last glacial maximum, approximately 20,000 to 26,000 years ago, was the most challenging period. Ice sheets covered Scandinavia, the British Isles, northern Germany, and Poland. Permafrost extended much further south than today. Sea levels were lower by more than 100 meters because so much water was locked up in glaciers, exposing continental shelves and land bridges. The climate was cold, dry, and harsh. Large parts of Europe that are forests today were tundra or steppe, supporting herds of reindeer, horses, bison, and mammoths rather than trees. Human populations contracted into the refugia we've discussed. The archaeological record from these refugia shows continued occupation and cultural development despite the harsh conditions. in the Iberian Peninsula. While we cannot directly test ancient blood types, researchers have developed indirect methods to infer genetic information from ancient remains. Ancient DNA analysis has revolutionized our understanding of prehistory over the past 2 decades. DNA can be extracted from bones and teeth, amplified and sequenced, revealing the genetic makeup of individuals who lived thousands or even tens of thousands of years ago. This technology has confirmed many hypotheses about prehistoric migrations and population relationships while overturning others. Several studies have examined ancient European DNA for blood group genes. One study of ancient Iberian samples found evidence of Rh-negative individuals in prehistoric populations, confirming that this trait is indeed ancient in this region. The frequency in ancient samples appears comparable to modern Basque frequencies, suggesting long-term stability. Other studies of ancient Europeans from various regions have also identified Rh-negative individuals, confirming the trait's presence in Paleolithic and Neolithic populations across the continent. These ancient DNA studies have also revealed that Neolithic farmers generally had lower frequencies of certain variants common in Paleolithic hunter-gatherers, consistent with the idea that incoming farmer populations diluted hunter-gatherer genetic variants in most of Europe, but less so in isolated areas like the Basque Country. As more ancient samples are tested, our understanding of how blood type frequencies have changed over time continues to improve. The archaeological record, combined with genetic analysis, paints a picture of complex population history in Europe, with multiple waves of migration, mixing, and cultural change over tens of thousands of years. The people carrying Rh-negative blood were hunters painting caves in Ice Age Europe, farmers in early agricultural villages, bronze workers and warriors, and eventually modern populations speaking Romance, Germanic, Slavic, and isolated non-Indo-European languages like Basque. Through all these transformations, this genetic variant persisted, carried by millions of individuals who likely never knew this distinctive feature of their blood, a feature that has become one of the enduring mysteries of human biology. The advent of modern genomics and computational biology has opened new chapters in our understanding of Rh-negative blood, though as with many scientific mysteries, new findings often raise as many questions as they answer. The late 20th and early 21st centuries have seen an explosion of genetic research, with technologies like DNA sequencing becoming faster, cheaper, and more accessible each year. These advances have allowed researchers to examine human genetic variation at unprecedented resolution and scale, revealing patterns that earlier scientists could only speculate about. The Human Genome Project, completed in 2003, provided the first complete sequence of human DNA, establishing a reference against which all human genetic variation could be compared. This monumental effort took 13 years and cost nearly $3 billion, but it laid the foundation for everything that followed. Today, complete genome sequencing can be done in hours for less than $1,000, and millions of people have had their DNA analyzed through commercial genetic testing services. These large-scale genetic data sets have enabled researchers to map human genetic variation across populations with extraordinary precision. Projects like the 1,000 Genomes Project, the Human Genome Diversity Project, and various national biobank efforts have sequenced DNA from tens of thousands to millions of individuals representing populations worldwide. These data sets allow researchers to identify rare variants, understand population relationships, trace migration patterns, and examine how natural selection has shaped human genomes. For Rh-negative blood, these modern studies have confirmed several key findings while revealing new complexities. Genetic analysis of the RHD gene region across populations has confirmed that the complete RHD gene deletion characteristic of most European Rh-negative individuals arose once and has been inherited from a common ancestor. By examining the genetic diversity surrounding the deletion, researchers have refined estimates of when this occurred, converging on dates between 30 and 50,000 years ago, though exact estimates vary depending on assumptions about mutation rates and demographic history. Studies have also revealed that while the complete Rhd deletion is the most common cause of Rh-negative status in Europeans, other genetic changes can produce the same phenotype. In African populations, several different Rhd variants have been identified that prevent normal D antigen expression. Some involve partial deletions, others have mutations that disrupt gene function, and still others involve complex genetic rearrangements. This genetic heterogeneity suggests that the selective pressure or drift that maintains Rh-negative blood might be operating similarly across different populations, even though the specific genetic changes differ. One particularly interesting finding from modern genetic studies concerns the relationship between Rh status and other genetic variants. Researchers have found that certain genetic variants nearby the Rhd gene are statistically associated with being Rh-negative, more than would be expected by chance. This suggests that the entire genetic region around Rhd might be under some form of selection, not just the D antigen itself. Whether this means nearby genes provide selective advantages that incidentally maintain the Rhd deletion, or whether there's some functional relationship between these genes remains unclear. Modern research has also examined whether there are health differences between Rh-positive and Rh-negative individuals beyond the well-known issues with pregnancy. Some studies have reported associations between Rh status and various conditions, though many of these findings have been inconsistent or controversial. For instance, some research has suggested Rh-negative individuals might have different risks for certain infections, autoimmune disorders, or even mental health conditions, but these associations are generally weak and not always replicated in different studies. One area that has received attention is the question of whether Rh-negative individuals have enhanced or reduced immune function compared to Rh-positive individuals. Given that the Rh protein is part of the red blood cell membrane and may interact with immune system components, it's plausible that Rh status could affect immune responses. Some studies have reported differences in antibody responses, cytokine production, or disease susceptibility based on Rh status. But again, findings have been inconsistent. The truth is probably that any health differences related to Rh status are subtle and context-dependent, making them difficult to detect and confirm. Evolutionary genetic analyses using modern genomic data have attempted to determine whether natural selection has acted on the Rhd gene region. Several statistical tests can detect signatures of selection in DNA sequences by looking for patterns that deviate from what would be expected under neutral evolution. Some studies applying these tests to the Rhd region have found weak signals that might indicate selection, while others have found no clear evidence. The lack of strong, consistent signals is itself interesting, suggesting that if selection is maintaining Rh-negative blood, it's likely through complex mechanisms involving multiple genes and environmental factors rather than simple directional selection favoring or disfavoring the trait. Researchers have also used computer simulations to model the evolution of Rh-negative blood under different scenarios. By creating virtual populations with specific parameters for population size, migration rates, selection coefficients, and genetic drift, they can see which scenarios produce patterns similar to what we observe in real populations. These models have shown that maintaining the observed frequencies of Rh-negative blood in European populations requires either very recent origin of the trait, some selective advantage that offsets the reproductive costs, or specific demographic scenarios involving population structure and isolation. Ancient DNA has provided direct evidence about the frequency of Rh-negative blood in prehistoric populations. As mentioned earlier, analysis of DNA from ancient skeletons has identified Rh-negative individuals in samples from across Europe and spanning thousands of years. Interestingly, some studies have suggested that the frequency of Rh-negative blood in ancient hunter-gatherer populations might have been higher than in modern European populations, which would fit with the idea that incoming farmer populations had lower frequencies and diluted the trait except in isolated refuges. One fascinating recent development is the use of ancient pathogen DNA to understand disease history. Researchers can extract and sequence DNA from bacteria and viruses preserved in ancient remains, revealing what diseases affected prehistoric populations. By comparing these pathogen genomes to modern strains and examining which human genetic variants are associated with resistance or susceptibility, scientists are reconstructing the coevolutionary history of humans and their diseases. While no specific pathogen resistance linked to Rh-negative blood has been definitively identified, this remains an active area of research. The molecular structure of the Rh proteins has been examined in detail using techniques like X-ray crystallography and cryo-electron microscopy, which can visualize proteins at atomic resolution. These studies have revealed that the Rh proteins have a complex structure with multiple membrane-spanning domains, creating a channel-like structure in the cell membrane. This structure supports the hypothesis that Rh proteins transport something across the membrane, though exactly what and under what circumstances remains debated. Functional studies in cellular and animal models have attempted to understand what Rh proteins do. Experiments deleting or altering Rh genes in mice, yeast, and bacteria have shown effects on growth, stress resistance, and gas transport. But translating these findings to humans is complicated by differences between species. Humans lacking the D antigen appear to have no obvious deficits in normal circumstances, suggesting either that the protein's function isn't essential or that other proteins can compensate for its absence. Researchers have also examined whether there might be subtle physiological differences between Rh-positive and Rh-negative individuals that don't manifest as overt disease but could affect fitness under certain conditions. For instance, some studies have looked at exercise physiology, cognitive function, stress responses, and metabolic parameters, comparing Rh-positive and Rh-negative subjects. Occasionally, small differences are reported, but these are typically modest and require replication before being considered reliable. The application of CRISPR gene editing technology has opened new possibilities for studying Rh genes. Researchers can now precisely modify genes in cultured cells, creating Rh-positive and Rh-negative cell lines that are otherwise genetically identical. This allows for controlled experiments to determine the exact consequences of having or lacking the D antigen, potentially revealing subtle effects that epidemiological studies might miss. Such research is still in relatively early stages, but it holds promise for finally determining whether the D antigen has functional significance beyond its role in immune recognition. International collaborative efforts have also advanced our understanding. Large-scale genetic association studies, where researchers examine hundreds of thousands or millions of individuals looking for connections between genetic variants and health outcomes, have included analysis of Rh status. These massive studies have the statistical power to detect even small effects that individual studies might miss. While they've confirmed the lack of major health consequences for Rh-negative status in modern populations with medical care, they continue to explore whether subtle associations exist with conditions ranging from infectious diseases to neurological disorders. One emerging area of interest is the potential role of Rh genes in neurological function. Some research has suggested that Rh proteins might be expressed in the brain or that blood-type antigens could influence brain chemistry through indirect mechanisms. Speculative connections have been made between Rh status and personality traits, cognitive styles, or susceptibility to certain mental health conditions, but these remain highly controversial and poorly supported. The brain is separated from the bloodstream by the blood-brain barrier, which would limit any direct effects of red blood cell antigens, though indirect effects through immune system interactions or shared genetic regulation aren't impossible. The relationship between Rh-negative blood and pregnancy continues to be studied from multiple angles. While Rho-D, immune globulin, has largely solved the clinical problem, researchers are working to understand the immunological mechanisms in greater detail. Why exactly do some Rh-negative women develop antibodies while others don't? What determines the severity of the fetal response? Can we predict which pregnancies will be most severely affected? Understanding these questions could lead to even better prevention and treatment strategies. There's also interest in whether the mechanisms involved in Rh incompatibility might shed light on other immunological problems, particularly other forms of fetal-maternal incompatibility and transplant rejection. The immune system's ability to recognize and respond to foreign antigens is central to medicine, and every additional understanding of how these processes work potentially has broader applications. Modern research has also explored whether historical patterns of Rh incompatibility might have influenced cultural practices. Some anthropologists and historians have speculated about whether societies might have developed taboos or preferences in mate selection that inadvertently reduced the risk of Rh problems, though direct evidence for this is lacking. Marriage patterns, birth spacing, and other cultural practices certainly could have influenced the reproductive consequences of Rh-negative blood, but demonstrating specific historical connections is extremely difficult. The development of artificial blood and blood substitutes has also touched on questions about Rh antigens. Researchers working to create synthetic blood products or to modify red blood cells for universal compatibility have had to grapple with the complexity of blood group antigens. Understanding exactly which antigens are essential and which can be safely removed or modified is crucial for these efforts. Looking forward, several promising research directions may help resolve remaining mysteries about Rh-negative blood. Larger ancient DNA datasets, as more samples are collected and sequenced, will provide better resolution of how Rh frequencies have changed over time and space. More sophisticated computational models of human demographic history will test competing scenarios about how the trait has been maintained. Continued functional studies of Rh proteins may finally reveal what they do and whether lacking them has any physiological consequences. And long-term epidemiological studies following large cohorts of people over years or decades may detect subtle health associations that smaller studies miss. Despite all these advances, the core mystery remains. We understand the genetics better, we know the trait is ancient, we've mapped its distribution precisely, and we've explored numerous hypotheses about its maintenance. Yet we still cannot definitively explain why Rh-negative blood exists at the frequencies we observe, particularly in populations like the Basques. This persistence of mystery in the face of intensive research is both frustrating and fascinating, a reminder that biological systems are complex and that human evolution involves countless interacting factors, many of which remain hidden from our current understanding. The story of Rh-negative blood is thus a story of ongoing scientific inquiry, of how researchers approach a problem from multiple angles, gradually accumulating knowledge while acknowledging the limits of what can be known. It exemplifies how modern biology works, combining genetics, evolution, anthropology, medicine, and computational analysis to address questions about who we are and where we came from. And it reminds us that sometimes the most interesting questions are those that don't have easy answers, that challenge our assumptions, and that keep us searching for deeper understanding. Beyond the well-documented challenges of hemolytic disease in pregnancy, the question of whether Rh-negative blood is associated with other health differences has long intrigued researchers and sparked considerable speculation. While some claimed associations have entered popular culture, the scientific evidence remains mixed and often controversial. Separating genuine biological effects from statistical artifacts, confounding variables, and the natural human tendency to find patterns even in random data requires careful analysis. The most rigorously studied health aspect of Rh-negative blood, apart from pregnancy complications, concerns infection and immune function. The immune system is extraordinarily complex, involving dozens of cell types, hundreds of signaling molecules, and intricate regulatory networks. Blood type antigens, including the Rh system, are part of this landscape, and it's plausible that they might influence immune responses in ways we don't fully understand. Some studies have examined whether Rh-negative individuals have different susceptibilities to infectious diseases. Research from the mid-20th century suggested that Rh-negative individuals might have lower rates of certain viral infections. including smallpox and polio, though these studies were small and not always well controlled. More recent research has looked at modern infectious diseases, including influenza, HIV, and various bacterial infections with inconsistent results. Some studies report small differences in infection rates or disease severity based on Rh status, while others find no association. One hypothesis is that the D antigen on red blood cells might serve as a receptor or co-receptor for certain pathogens, meaning that lacking it could provide protection against those specific infections. However, identifying such pathogens has proven difficult. No major human pathogen has been definitively shown to require the D antigen for infection, though the possibility remains that some minor or regional pathogens might be affected. Conversely, some research has suggested that Rh-negative individuals might have enhanced immune responses in certain contexts, producing more antibodies or having more active inflammatory responses. This could theoretically provide advantages against infections but might also increase risk for autoimmune conditions where the immune system attacks the body's own tissues. Indeed, some studies have reported associations between Rh-negative blood and higher rates of conditions like rheumatoid arthritis, multiple sclerosis, or psoriasis, though again, findings have been inconsistent across studies. The relationship between blood type and disease is complicated by the fact that many diseases have complex causes involving multiple genes, environmental factors, and gene-environment interactions. Detecting a modest effect of Rh status against this noisy background requires very large studies with careful controls for confounding variables. Many older studies of blood type and disease were too small or methodologically flawed to provide reliable evidence, and even modern studies sometimes produce conflicting results. One area where some evidence has accumulated concerns toxoplasmosis, an infection caused by the parasite Toxoplasma gondii. This single-celled parasite infects a wide range of mammals and birds, with cats serving as its primary host. Humans usually acquire it from undercooked meat or contact with cat feces. Most infections are asymptomatic or cause only mild flu-like illness, but the parasite can establish chronic infection in the brain and other tissues. Research has suggested that toxoplasma infection might influence human behavior in subtle ways, affecting personality, reaction times, and even mental health, though these findings remain controversial. Several studies have reported associations between Rh-negative blood and different responses to toxoplasma infection. Some research suggests Rh-negative individuals might be more susceptible to infection or might experience different behavioral effects, though the mechanisms and reliability of these associations are debated. If true, this could represent a genuine biological interaction between Rh status and pathogen response, but much more research would be needed to confirm and understand such an effect. Cancer has also been investigated for potential associations with Rh status. Various studies have examined whether Rh-negative individuals have different risks for different types of cancer, with most finding no strong associations. Some research has suggested possible links between Rh-negative blood and certain reproductive cancers or differences in cancer survival rates, but these findings haven't been consistently replicated. Given that cancer comprises hundreds of distinct diseases with different causes, it would be surprising to find a universal effect of Rh status, though specific associations with particular cancer types remain possible. Cardiovascular disease, the leading cause of death in most developed countries, has been examined for blood type associations. Research has established that ABO blood type influences cardiovascular risk, with type O individuals having somewhat lower risk than other types, possibly related to effects on blood clotting and inflammation. The relationship between Rh status and cardiovascular disease is less clear. Some studies have suggested Rh-negative individuals might have slightly different risks for certain cardiovascular conditions, but evidence is weak and inconsistent. Neurological and psychiatric conditions have attracted particular interest regarding Rh associations, partly because some studies have reported intriguing correlations and partly because speculation about Rh-negative blood often touches on psychological differences. Research has examined schizophrenia, depression, autism spectrum disorders, ADHD, and various other conditions, looking for associations with Rh status. A few studies have reported correlations, but the majority have found no significant associations, and those that do report findings often conflict with each other. The speculation about psychological differences in Rh-negative individuals extends into popular culture, with claims about enhanced intuition, psychic abilities, higher intelligence, or different personality traits. These claims are not supported by rigorous scientific evidence. While it's theoretically possible that Rh status could influence brain development or function through indirect mechanisms, no plausible biological pathway has been identified, and careful psychological studies have not found consistent differences in cognitive abilities or personality traits based on Rh status. Allergy and asthma have also been investigated. The immune dysregulation that leads to allergic conditions could potentially be influenced by factors affecting immune function, including theoretically blood-type antigens. Some research has suggested Rh-negative individuals might have different rates of certain allergies, but findings are inconsistent and the mechanisms unclear. Pregnancy outcomes beyond hemolytic disease have been studied as well. Research has examined whether Rh-negative women have different risks for preeclampsia, gestational diabetes, preterm birth, or other pregnancy complications with mixed results. Some studies suggest possible associations, while others find none. The complexity of pregnancy, involving the interaction of maternal, fetal, and placental factors, makes detecting subtle effects of any single variable challenging. One interesting line of research has examined longevity and aging. Do Rh-negative individuals live longer or shorter lives on average? Do they age differently? Studies addressing these questions have generally found no significant differences in lifespan based on Rh status alone, though some research has suggested possible interactions with other factors. For instance, the health advantages or disadvantages of being Rh-negative might differ depending on environmental conditions, lifestyle factors, or other genetic variants. The challenge in all this research is distinguishing genuine biological associations from statistical noise and confounding variables. When researchers test dozens or hundreds of potential disease associations, some will appear statistically significant by chance alone. This is the problem of multiple comparisons. Additionally, populations differ in many ways beyond just Rh status. If Rh-negative individuals are more common in certain ethnic groups, and those groups have different disease rates for historical, cultural, or environmental reasons, an association might appear between Rh status and disease that isn't actually causal. To establish that Rh status genuinely influences a health outcome requires several types of evidence. The association should be replicated in multiple independent studies, ideally in different populations. There should be a plausible biological mechanism explaining how Rh status could affect the condition. The association should remain significant after controlling for potential confounding variables. And ideally, experimental studies in cellular or animal models should demonstrate the proposed mechanism. For most claimed health associations with Rh-negative blood, these criteria are not met. The associations tend to be weak, inconsistently replicated, lacking clear mechanisms, and often confounded by population structure. This doesn't mean there are definitely no health effects of Rh status beyond pregnancy complications, but it does mean we should be cautious about accepting claimed associations without strong evidence. It's worth noting that even small health effects could be evolutionarily significant. A trait that increases survival or reproduction by even 1 or 2% can be strongly selected over many generations. So if Rh-negative blood provides even modest advantages in certain contexts, this could help explain its persistence. The difficulty is detecting such modest effects reliably, especially for complex traits influenced by many factors. Another consideration is that health effects might be context dependent. Perhaps being Rh negative provides advantages under some conditions but disadvantages under others. Or perhaps effects differ depending on other genetic variants an individual carries. Modern genomics is revealing that most health outcomes involve complex interactions among many genes and environmental factors. Rh status might be one piece of a larger puzzle, with effects that are only apparent in specific genetic or environmental contexts. The persistence of interest in health associations with Rh-negative blood reflects both legitimate scientific curiosity and the human tendency to seek explanations and patterns. People who are Rh-negative or have Rh-negative family members naturally wonder whether this trait might influence other aspects of health or biology. Researchers continue to investigate because even if most associations turn out to be spurious, discovering a genuine one could provide important insights into immunology, evolution, or medicine. What we can say with confidence is that in modern populations with access to medical care, being Rh negative doesn't dramatically affect health outside of pregnancy management. Rh negative individuals live normal lifespans, aren't dramatically more or less healthy than Rh positive individuals, and don't require any special medical management beyond pregnancy precautions. This is reassuring for the millions of people with this blood type, even as the broader mystery of why the trait exists continues to intrigue researchers. Beyond the scientific questions, Rh-negative blood has captured popular imagination in ways that few other genetic traits have. This cultural phenomenon itself is interesting, revealing something about how humans relate to biological mysteries and what we find compelling about our own nature and origins. The story of Rh-negative blood has spilled out of scientific journals into popular books, documentaries, internet forums, and countless conversations, taking on a life that often bears little resemblance to the careful, uncertain conclusions of actual research. Part of the fascination undoubtedly stems from the trait's mysterious nature. Here is something that millions of people carry in their blood, something that can be precisely measured and tested, yet something we cannot fully explain. In an age when science seems to have answers for everything, when we can sequence entire genomes and edit genes with precision, the persistence of this unexplained biological feature is compelling. It reminds us that nature still holds secrets, that we don't understand everything about ourselves, that reality can still surprise us. The geographic concentration of Rh-negative blood, particularly in the Basque Country, adds to the intrigue. The Basques themselves are mysterious, with their ancient language, genetic distinctiveness, and cultural persistence. When you combine an enigmatic people with an enigmatic trait they carry in high frequency, the result is irresistible to those interested in human origins and prehistory. It suggests connections to the deep past, to populations and events we can only dimly perceive through fragmentary evidence. This combination of mystery and specificity has made Rh-negative blood fertile ground for speculation and alternative theories. Some of these theories remain within the bounds of scientific plausibility, proposing connections to ancient populations, unknown selective pressures, or complex demographic history. Others venture into more speculative territory, suggesting connections to lost civilizations, ancient aliens, or mystical properties. While these latter theories lack scientific support, their popularity reveals the human need for narratives that explain our origins and make us feel connected to something larger and more mysterious than everyday existence. The internet has amplified these discussions, creating communities of people interested in Rh-negative blood, sharing experiences, theories, and speculations. Some Rh-negative individuals report feeling different or special, experiencing unusual health patterns, having vivid dreams or intuitions, or feeling disconnected from mainstream society. While these subjective experiences are real to those who report them, they're difficult to study scientifically. Self-selection bias, where people with unusual experiences are more likely to join such communities and share their stories, can create an impression of distinctiveness that might not exist in the broader population of Rh-negative individuals. The tendency to ascribe special significance to Rh-negative blood can be understood psychologically. Humans are pattern-seeking creatures. We evolved to detect meaningful patterns in our environment, to find causes for effects, to construct narratives that make sense of experience. This cognitive trait serves us well in many contexts, but can also lead us to perceive patterns that aren't really there or to overinterpret genuine but modest biological differences. Additionally, people naturally seek identity and belonging. Discovering that you carry a relatively rare genetic trait can become part of your self-concept, a way of understanding yourself as unique or connected to a particular heritage. This isn't harmful in itself and may even be psychologically beneficial, providing a sense of identity and connection to history. The danger comes only when speculation becomes untethered from evidence, when extraordinary claims are accepted without extraordinary proof, or when biological differences are used to create hierarchies or divisions among people. From a scientific standpoint, the appropriate response to the mystery of Rh-negative blood is neither to dismiss it as unimportant nor to accept wild speculations uncritically, but rather to maintain what one might call informed curiosity. Yes, this is a genuine biological mystery. Yes, we don't fully understand it. Yes, further research might reveal important insights about human evolution, population history, or immunology. But no, the lack of complete explanation doesn't mean we should abandon scientific standards of evidence or accept explanations that contradict established knowledge without compelling proof. The story of Rh-negative blood also illustrates how scientific understanding develops over time. From the initial discovery of blood groups, to the identification of the Rh factor, to the development of treatments for hemolytic disease, to modern genetic and evolutionary analyses, our knowledge has grown progressively. Each generation of researchers has built on previous work, correcting errors, refining theories, and asking new questions. This is how science works, not through sudden revelations or complete understanding, but through gradual accumulation of evidence and iterative refinement of ideas. The cultural fascination with Rh-negative blood may also reflect broader questions about identity and origins that resonate deeply with people. Were we? Where did we come from? What makes us unique? These are fundamental human questions, asked in every culture and every generation. A genetic trait that connects some people to ancient populations, that shows clear geographic patterns, that seems to tell a story about human prehistory, becomes a vehicle for exploring these larger questions. For those who carry Rh-negative blood, learning about their blood type can be a moment of connection to the deep past, a tangible link to ancestors who lived through the Ice Age, who painted caves, who survived in mountain refuges when glaciers covered much of Europe. It's a reminder that we are not separate from nature or history, but participants in an ongoing evolutionary story that stretches back hundreds of thousands of years and will continue long into the future. For researchers, the mystery of Rh-negative blood represents both a challenge and an opportunity. The challenge is to explain how and why this trait has persisted despite apparent disadvantages. The opportunity is that solving this puzzle might reveal important principles about human evolution, population genetics, or selection mechanisms that apply more broadly. Every mystery solved tends to illuminate not just the specific question, but to shed light on related issues and generate new questions for investigation. The persistence of Rh-negative blood as a topic of popular interest, appearing regularly in documentaries, books, and online discussions, ensures that research in this area receives attention and potentially funding. Public interest can drive scientific inquiry, encouraging researchers to pursue questions they might otherwise neglect. At the same time, researchers must be careful not to let popular expectations or desires for sensational findings influence their work. The goal must remain finding truth through rigorous methodology, even if the truth turns out to be more mundane than exotic theories suggest. What seems clear is that Rh-negative blood will continue to fascinate people as long as its evolutionary story remains incompletely understood. Today we can definitively explain how this trait arose, why it persisted, and what advantages or disadvantages it confers. Much of the mystery will dissipate. Until then, it remains a genuine biological puzzle, a reminder that our bodies carry histories written in molecules, stories we're still learning to read. The broader lesson might be that science is not just about what we know, but about how we approach what we don't know. Do we respond to uncertainty with wild speculation, with dismissal, or with careful, systematic investigation? Do we maintain both open-mindedness to new ideas and critical thinking about claims? Do we accept that some questions might take generations to answer fully? The story of Rh-negative blood teaches us to embrace mystery while pursuing understanding, to be comfortable with uncertainty while working to reduce it, and to appreciate both the progress we've made and the vast frontiers that remain unexplored. As we draw our journey through the mystery of Rh-negative blood toward its close, we find ourselves in a curious position. We've traveled through centuries of scientific discovery, from Carl Landsteiner's groundbreaking identification of blood types to modern genetic sequencing that can read our molecular histories. We've explored the peculiar distribution of this trait across the globe, its extraordinary concentration among the Basques, and the evolutionary paradox it presents. We've delved into theories of ancient origins, examined the archaeological record, and considered modern research findings. Yet despite all this investigation, all this accumulated knowledge, the central mystery remains unsolved. Why does Rh-negative blood exist at the frequencies we observe? This simple question, asked decades ago and still asked today, awaits a definitive answer. We have pieces of the puzzle. We know the trait is ancient, likely tens of thousands of years old. We know it's concentrated in European populations, particularly in southwestern Europe. We know it presents reproductive disadvantages before medical intervention. We know it's maintained by some combination of selection, drift, migration, and population history. But we cannot yet assemble these pieces into a complete, satisfying picture that explains everything we observe. Perhaps this is how it should be. Science is not a collection of facts but a process of inquiry, and the most interesting questions are often those that resist easy answers. The mystery of Rh-negative blood has driven research for more than 80 years, motivating studies in genetics, immunology, anthropology, and evolutionary biology. It has led to life-saving medical treatments, deepened our understanding of human prehistory, and revealed unexpected complexity in human genetic variation. If the mystery were simple, if the answer were obvious, we might have learned less in the process of investigating it. For the millions of people around the world who carry Rh-negative blood in their veins, this trait is simply part of who they are, as unremarkable as eye color or height. They live ordinary lives, experiencing the same joys and sorrows, challenges and triumphs as anyone else. The Rh-negative status matters primarily during pregnancy, when modern medicine can manage the risks effectively and otherwise fades into the background of daily existence. Yet beneath this ordinariness flows an extraordinary history, a genetic legacy connecting them to ancient populations, to prehistoric migrations, to the deep currents of human evolution. The Basques, carrying the highest concentration of this trait, continue their lives in the mountains and valleys of the Western Pyrenees, speaking their ancient language, maintaining their cultural traditions while also participating fully in modern European society. Their blood carries a molecular memory of ice ages and ancient refuges, of populations that survived when others perished, of genetic continuity spanning countless generations. They are both unique and ordinary, simultaneously a living link to the Paleolithic past and contemporary people navigating 21st century challenges. Research into Rh-negative blood continues in laboratories around the world. Geneticists sequence more ancient DNA, adding to our knowledge of prehistoric populations. Evolutionary biologists refine their models, testing new hypotheses about selection and drift. Immunologists investigate the molecular details of Rh proteins, seeking to understand their function. Epidemiologists analyze large data sets, looking for health associations. Each study adds another small piece to the puzzle, incrementally advancing our understanding. Perhaps someday a breakthrough will come. Perhaps ancient DNA from a crucial time period will reveal when and how Rh-negative frequencies changed. Perhaps a large-scale health study will identify a clear advantage to being Rh-negative that explains its persistence. Perhaps new evolutionary models will show how the observed patterns could arise from previously unconsidered demographic scenarios. Or perhaps the answer will come from an unexpected direction, from research into seemingly unrelated topics that suddenly illuminates this old mystery. Or perhaps the mystery will persist for generations more, continuing to intrigue and puzzle researchers and laypeople alike. Science has no obligation to provide answers on our preferred timeline. Nature reveals its secrets when and how it chooses, through the patient accumulation of evidence and the occasional flash of insight. Some questions are answered quickly. Others require centuries of investigation. The satisfaction comes not just from the final answer, but from the journey of discovery, from each new piece of understanding, from the gradual refinement of our picture of reality. What we can say with certainty is that human beings are endlessly complex, shaped by millions of years of evolution, thousands of generations of history, countless migrations and mixings and isolations. We carry in our bodies the traces of all this history, written in molecular codes we're only beginning to fully decipher. Every person is a biological archive, a living fossil, a story told in proteins and DNA. Rh-negative blood is one chapter in this larger story, a particularly puzzling chapter, but just one among many. The pursuit of knowledge about our origins and nature is fundamentally human. We alone among Earth species ask where we came from, how we became what we are, what makes us distinctive. We study our own biology with the same curiosity we bring to studying stars, atoms, and distant galaxies. And in doing so, we discover that we are both special and ordinary, both unique and connected to all life, both deeply understood and still profoundly mysterious. The story of Rh-negative blood teaches us humility in the face of nature's complexity. It reminds us that obvious questions can have subtle answers, that biological systems involve countless interacting factors, that evolution operates through mechanisms we're still working to fully understand. It shows us that human prehistory was more complex than simple narratives suggest, involving population movements, isolations, mixings, and survivals that left traces in our genes. It also teaches us the value of patience in scientific inquiry. The researchers who first identified the Rh factor could not have imagined the sophisticated genetic analyses we conduct today. The scientists working today cannot fully anticipate what tools and techniques future researchers will have at their disposal. Each generation does the best work it can with available methods, knowing that future generations will build upon and perhaps revise their conclusions. This is not a failing of science, but its greatest strength, its self-correcting nature, its commitment to following evidence wherever it leads. For those who have followed this journey through the mystery of Rh-negative blood, the story offers no neat conclusion, no satisfying resolution where all questions are answered. Instead, it offers something perhaps more valuable, an appreciation for the complexity of nature, an understanding of how science approaches mysteries, and a sense of connection to the deep human past. It invites us to see ourselves as participants in an ongoing evolutionary story, carrying traits and genes inherited from ancestors who survived ice ages, migrations, plagues, and countless other challenges. As you settle into sleep tonight, as your mind drifts toward rest, perhaps you'll think about the blood flowing through your veins, carrying oxygen to every cell, sustaining the life and consciousness that makes you who you are. Whether you're Rh positive or Rh negative, that blood connects you to billions of other humans, to ancestors stretching back through countless generations, to the very origins of our species in Africa hundreds of thousands of years ago. The mystery of Rh-negative blood is ultimately a mystery about us, about human diversity, about where we came from and how we became who we are. It's a reminder that even in our own bodies, even in something as seemingly simple as blood, profound questions await answers. And it's an invitation to embrace mystery as part of the human experience, to pursue understanding while accepting that some questions take time to answer, and to find wonder in the fact that we can ask such questions at all. Sleep well, knowing that science continues its patient work, that researchers around the world pursue these questions, adding piece by piece to our understanding. Sleep well, knowing that you carry within you a history deeper than any written record, a biological archive connecting you to the first humans and beyond. Sleep well, knowing that mystery and understanding can coexist, that unanswered questions are invitations rather than obstacles, and that the pursuit of knowledge is one of humanity's most noble endeavors. And if you have Rh-negative blood flowing through your veins, know that you carry a genetic variant with a fascinating story, a trait that connects you to ancient European populations, to mountain refuges during ice ages, to the Basque country and its enduring culture. You are part of the mystery, a living chapter in this ongoing story. But know also that you are, above all, simply human, sharing with all people the fundamental qualities that define our species: curiosity, creativity, resilience, and the capacity to wonder at the mysteries that surround and constitute us. The story continues. The research goes on. The mystery for now remains. And perhaps that's exactly as it should be. Thank you for joining me on this exploration of one of biology's most enduring mysteries. If you've enjoyed this journey through the enigma of Rh-negative blood, please consider subscribing to our channel for more fascinating explorations of history, science, and the hidden stories written in our genes. Give this video a thumbs up if you'd like to see more deep dives into topics that blend history, biology, and mystery. And let me know in the comments what other scientific mysteries you'd like to explore in future episodes. Until next time, sleep well, dream peacefully, and remember that you are part of a story millions of years in the making, carrying within you the history of our species and the mysteries that still await understanding. Good night.